Thank you for having me here. So I'm a PhD student at Sorbonne University in Paris, and I will talk to you about some of the work that I have done during my PhD, which is about trying to predict motions of proteins. Okay, so protein motions are important because they are relevant to the protein function. As you may know, in a biological environment, a protein may change its conformation in order to perform its function, whether it is to bind a certain partner or to form a complex. The change of conformation can modulate the activity of the protein. So the main question that we will ask during this presentation is how to leverage the sparse observations that are available in the PDB in order to study protein motions. So in order to study protein motions, we have created a pipeline and applied it at the PDB scale. So what it does is the following. First, it extracts all the chains available in a database. Then it clusters them using a user-defined level of sequence identity and coverage. Then it calls a multiple sequence alignment software on all the previously created clusters. And finally, using the sequence alignment information and the structural information, it performs structural alignment on a chosen reference, generating a conformational collection. So this whole process takes around 2 hours and 15 minutes to compute on a desk computer, and it generates around 35,000 collections. So in the paper, we have explored 8 different combinations of sequence identity and coverage. So I give here the size difference in the different collections between the most stringent setup, which is 80% identity, 80% coverage, and the most permissive one, which is 30% identity and 50% coverage. So what we see is that around half of the ensemble are not affected by the different clustering levels, but the ones that are affected tend to be the cluster containing the most studied protein in the PDB with a lot of resolved 3D structure. So DENSE is a tool that allowed us to build conformational collections of proteins. And now what we want to do is to extract from this conformational diversity what we call linear motions. So we want to do this because it's a compact representation of the conformational diversity. So we do that by diagonalizing the covariance matrix of the 3D coordinates of the conformations. And the eigenvector that we get in this process is interpreted as a linear motion. And the corresponding eigenvalue gives us the amount of variance explained by these linear motions. So to illustrate, I give on the left all the conformations of a given collection. Here in yellow, you have the first principal component, and here is the trajectory that we get when we follow that yellow path. So to give an idea about the complexity of the motion that we found in this database, we report for each one of the collection the number of principal components that are necessary to explain 90% of the positional variance. And what we see here is that around half of the ensemble only need 1 or 2 linear motion to explain almost half of their diversity, and that almost all of the ensemble need less than 10 modes to explain their diversity. I show here 2 examples, one with a very simple unimodal motion with adenylated kinase, and one with a more complex motion that need 3 linear modes to explain its diversity with ATPase. So since protein conformational diversity lie on a low-dimensional manifold, we wanted to assess the capacity of popular manifold learning techniques in the task of reconstructing unseen protein from a low-dimensional space. So we have selected 3 different meaningful learning techniques. So the first one is principal component analysis. Then we also have seen we have used kernel PCA and UMAP, and we have trained them on 10 different collections with different complexity of motions. We have trained them using leave one out procedure. So this means that we have clustered all the conformation inside a collection, and that we have trained on all the subcluster except one, and we have repeated this process until each one of the cluster has been seen as a validation cluster. So about the results we see that UMAP consistently perform the worst in blue-green, and that we can get high-quality reconstruction using PCA and kernel PCA. But what we also see is that this quality of reconstruction is strongly dependent on the distance to the training set. So now I want to present Simoon, which is a sequence-to-motion model. So the idea here is to leverage the database of motion that we have built using the dense tool that I have presented earlier. So what we do is that we start from protein per residue embeddings, and we try to decode them into linear motions using a neural network. And what we try to minimize is the normalized sum of square error between the prediction and the ground truth. We wanted to compare our purely sequence-to-motion approach with a physical approach called normal mode analysis. So normal mode analysis rely entirely on the structure. It represents the structure as an elastic networks, and it sees how this elastic networks oscillates around an equilibrium state. So I give here a quantitative assessment of our approach. So it's the number of test samples that are found below a certain threshold in loss value. And what we see is that our purely sequence-to-motion approach in blue performs similarly to the normal mode analysis. And when we use the PROST5 embeddings that are more structural aware than the other, we get the best performances. I want to show one example. So it's this example is better predicted by our approach than normal mode analysis. So it's the large unfolding motion of the anthrax protective antigen. So these two structures are found in the PDB. The motion that we have extracted is the yellow one. So we see that we get it using our approach, while the normal mode analysis struggle to get it because it tries to predict an opening and closing motion. So to conclude, we have seen that conformational diversity of a protein family lie on a low-dimensional manifold. We have seen that using classical manifold learning method, we can reconstruct unseen structure, but only in a closed extrapolation regime. And finally, I have presented an approach using protein language models to predict linear motions. I would like to thank you for your attention and my supervisor for their help. And if you have any question, I will be glad to try to answer.
Thanks, Valentin, for your nice talk. We have time for one question.
Do you use a lot of NMR structures, because they usually have collections of maybe like 30? And then when you do that, do you see more of like the disordered structure? And how does that like compare to the examples that you shown here?
So yes, thank you for your question. So we use an NMR structure. We have the possibility to use it or not in the pipeline. And how does it compare? We have not specifically took a look at this, so I cannot really comment on that.